Good afternoon. Welcome to this seminar this afternoon. Mr. R. Purushottam, the PhD student, will be presenting his seminar on ascertaining the extent of contribution of various traits to terminal drought tolerance in chickpea. There were certain uncertainties, and then the supervisor had been shifting between me, L. Krishnamurthy, and Vincent Vadas for a while. So ultimately I am here to introduce him. The topic had been a very tough one, and then which involves a lot of hitting the ground. So no one was interested in taking this up. But the topic was very important for building up the confidence of breeders so that they can take up various traits and consider them in their breeding programs. Purushottam joined in October 2010, and he had just worked on this topic since then, most of the time in the field. And then since 9 months, he started writing his papers and thesis. So I would ask Mr. Purushottam to present his work.
A very good afternoon to everyone. I am R. Purushottam and I am going to present my PhD work entitled Ascertaining the Extent of Contribution of Various Traits to Terminal Drought Tolerance in Chickpea. Before getting into that, you will see the title itself is all to validate the trait that contributes to drought tolerant. At this point, I wish to mention one issue. The topic chosen was difficult, as my supervisor said, and demands enormous collection of data. Therefore, I am not in the position to present all the data, but I'll just touch upon the introduction, methods used, and present a sample of primary data, but I will elaborate the association and relationship and discussion. Similarly, in objective 2 and 3, I will also briefly explain the methods used and the observation was measured, but I will elaborately discuss about the discussion and results. While coming to introduction, chickpea is the third most important grain legumes and majorly grown in South Asia, West Asia and North Africa and Mediterranean region. It is also known as high nutritive protein crop, and chickpea is usually grown under stored soil moisture with minimal rainfall or no rainfall during the cropping season. At this cropping season, there is a constant decline of soil water, resulting in a terminal drought stress. When taking up the drought tolerant breeding, the constitutive trait or trait that is well associated to grain yield rather than the grain yield itself is suggested to be select for. As everybody knows, the grain yield is prone to G into E interaction. Coming to that, the trait proposed to associate with the grain yields are many in numbers. For example, 17 root related traits were proposed. It is a sample trait, it's not everything. There are 17 root related traits and 16 shoot related traits, and 8 yield components were shown in this slide. But we all know those traits are important, but there is a pressing need to suggest the critical order of the trait to drought tolerance. Integration of traits for drought tolerance is not only we come to know that mechanism as well as the type and intensity of root, we also need to understand the traits involved and their mechanisms. There are two different models. It's not entirely different, but similar models on water-related crop components. There were totally six in number, which is called as composite traits, which is really helpful for selecting the drought tolerance in chickpea. The objective of my study is to understand the relative importance of various traits which is contribute to the grain yield, and also study the marker diversity within that studied genotype. The second objective deals with the evaluate the CTD as a trait and the stage of relevance and finding a possible marker trait association for CTD. Then the third objective is deal with to compare the root anatomy of chickpea with the other grain legumes, and also between the types of chickpea to understand the axial resistance of soil water uptake. While entering into objective, the trial was conducted in RCBD with 3 replication with 2 different soil water treatment. One is non-irrigated and the other one is optimally irrigated. The sowing date was 30th October 2009 in the first year, but it delayed to 20th November 2010. The selection, you can see there are 12 number of genotypes which is used in this genotype, but I want to clearly mention that this genotype selection was not random. There is a purpose of selection for these 12 genotypes. The first 2 genotypes, as you see, the first 2 genotypes are very strong in their root system at the vegetative stage, and these 1882 and 283 for their weak root system at the vegetative stage as well. And also we have selected 2 drought tolerant lines, field-based drought tolerant genotypes shown a highly tolerant reaction in field, and also ICC 3325 and 14799 they showed a low level of tolerance in the field condition. And the genotypes 3776 and 7184 were selected for their sensitivity. As a check, we selected Anigiri and ICCV 10 to compare those genotypes with the local adapted variety. The measurements were taken was roots related traits, shoot related traits, soil moisture observation, and canopy temperature, especially at the mid reproductive stage. The grain yield and shoot biomass, harvest index, drought tolerance index, and morphological and analytical yield components were measured at maturity. The root system were measured using soil core method, and the soil moisture were observed using TDR probe meter. The drought tolerance was measured as a grain yield. If required, we can consider in this study, it also considered the drought tolerance index and shoot biomass production. The objective of the study is focused mainly on the relationship between traits and their contribution to yield. Therefore, we performed correlation, regression, and mainly path coefficient analysis to dissect it very finer of their contribution levels. And also we had a genetic diversity analysis of this studied genotype. Both SNP, DOT and SSR markers were analyzed using power marker version, and the dendrogram tree was constructed using Darwin, Antisys and Meho software. The results, I'm sorry for the crowded data. This is primarily to show the pattern of root traits that we have collected in the field is mainly deals with root length density and root dry weight for all the 12 genotypes in both depth levels in the sense it each 15 centimeter, and their mean total were also considered for this study in both non-irrigated and irrigated condition. As well, the crop utilized soil moisture of 12 diverse genotype was also measured in both the condition. It also measured up to 120 centimeter. In the later stage, you can see this was measured at 75 days after sowing. The samples were taken at this stage was 120, but at the initial stages it was considered only up to 60 because the rooting extent of the chickpea genotypes at flowering stage is only the maximum of 60 at the drought condition. So in those early growth duration, we consider only 60 and it keep on increasing up to 120 based on their rooting instant and as well the crop growth stage. Likewise, we have a shoot data which is clearly focused on this shoot biomass production, SLA and LAI. Those different components of shoot biomass were quantitatively calculated and then the SLA and LAI for all the genotypes. Likewise, canopy proportion and canopy temperature were also measured for those 12 genotypes. The canopy proportion is calculated by the canopy area to the ratio between the ratio of canopy area to that of ground level area. For the canopy temperature depression is the difference between the air temperature and the canopy temperature. Likewise, we have the phenology, shoot biomass, grain yield, harvest index and drought tolerance index for the 12 diverse genotypes. In this case, I want to clearly mention these ICC 4958 and 8261 were produced a large root system, as I already mentioned, but while in phenology, ICC 4958 and Anigri were earliest in duration, and the small root producing weak root system genotypes 1882 and 283 were little longer than the early duration genotypes. The remaining genotypes were medium in duration. When come to grain yield production, the genotypes ICC 4958 and the genotypes that are highly drought tolerant ICC 867 and 14778 and the best adapted varieties Anigri and ICC with 10 were yielded high, but the weak root genotypes 1882 and 283 were yielded poor. The small roots were yielded moderate, but the drought sensitive genotypes 3776 and 7184 were yielded very poor under drought condition. The increase in shoot biomass didn't reflect in the yield increase at this stage, but harvest index reflect in the grain yield increase. In this year, there was a rain received immediately after the last irrigation in the optimally irrigated condition, where caused the excessive shoot biomass production in the optimally irrigated condition, resulting in a poor harvest index and grain yield compared to the drought condition. Though there were a problem, the drought tolerant index can able to discriminate their tolerant genotypes with the sensitive genotypes. We'll come to morphological and analytical yield components. The pod number and seed number were reflect on the grain yield increase, except that large seeded genotype 4958 and 8261, and the seed number and 100 seed weight were clearly discriminate the genotypes variation. While coming to the analytical yield components, the partitioning coefficient is explained very well the genotype discrimination compared to the crop growth rate and DR at this stage. The relationship between the root proliferation and the root proliferation at a certain soil zone and to the amount of soil water uptake for that particular zone is expected to be linearly related. In this case, I just want to show the RLD of the particular depths with the crop utilized soil moisture for that depth where regressed. You can find in the soil clear non-association in the initial surface layer at 80 days after sowing, but generally it was like it starts from 35 days itself. And also the freshly descendant rooting depth also doesn't contribute, or else otherwise the freshly extended root zones were not significantly related with the rootland density and crop utilized soil moisture. You can see the clear pattern in this 2010-11 data. The soil surface area doesn't showed a non-significant relationship between rootland density and crop utilized soil moisture, as well the freshly extended zone. These kind of data are very rare in literature, and this set of data is from both the years, showing their importance of soil water uptake by RLD. The summary of root traits is mainly deal with the beginning of flowering as well as at the initiation of reproductive stage. The root depth was the maximum at 60 centimeter. At this stage, drought stress enhanced the root dry weight as well as rootland density in 2010-11. Based on that, generally all the 12 genotype fell into major 4 categories, which is highly root producing genotypes at the vegetative stage have a good vegetative stage root growth, and the highly drought tolerant lines ICC 867 and 14778 and the tolerant lines were, and also the weak root producing genotype at the initial stage had a very good reproductive stage root growth, and the highly tolerant lines and the best adapted varieties were able to maintain the root growth across stages. And the last group is consists of the drought sensitive genotypes, which is not performed very well in root growth across stages. While talking about the crop utilized soil moisture, the drought stress reduced the soil moisture uptake in drought stress compared to the optimally irrigated condition. ICC 4958 and 3325 at vegetative stage, and the best adapted variety ICC 10 and 14 highly tolerant genotype 14778 were utilized the maximum amount of water at the reproductive stage, and the genotypes Anigiri and 14779 had a high soil moisture utilization across stages. But similarly, we have seen the root growth of 3776 and 7188. They were very poor in root growth as well soil water utilization. When come to the summary of shoot traits, ICC 4958 and 8261, the best adapted varieties were relatively early in shoot biomass production, though ICCV 10 were delayed at that time, catching up their shoot biomass production at the reproductive stage. At early vegetative stage, shoot biomass production and LAI did not discriminate the highly drought tolerant lines and drought tolerant lines with the sensitive one, but the highly tolerant lines and the best adapted varieties were able to pick up their the highly drought tolerant lines and the drought tolerant lines were able to maintain their shoot biomass production a level up in the reproductive stage. This yield components is mainly discriminated. The pod, as I already mentioned, the pod number and the 100 seed weight were reflected in the grain yield and showed a highest genetic variation compared to the drought tolerant index. The genotype distribution for 100 seed weight was directly inverse to the pod number per meter square. 100 seed weight of genotype ICC 4958, 8261 and the best adapted varieties were greater than the mean across stages in both the years also. When we come to analytical components, irrigation extended DV but not, and also the DR, but the DR in an extended manner. The late duration genotype had a highest DV but reduced in DR. Till now we have see the pattern of shoot growth, root growth and soil moisture utilization across the 12 genotypes in both the irrigated and non-irrigated condition, and these slides is represent to dissect the contribution of root traits, the yield. The path coefficient analysis had advantage to dissect the correlation into direct and indirect effect. In this case, the embolded ones in diagonal manner are the direct effect of these traits, and the values here under the yield are the correlation coefficient of yield and these particular traits respectively. At 35 days after sowing, the root present at the initial stages was not significantly contribute, but it shows the presence of contribution to the yield in the 35 days after sowing. In both root dry weight, it also shows the positive contribution and also have a direct positive effect in all the cases except 45 to 60 depth. The same data was also measured at the flowering stage as well as the pod initiation stage in both the years. You can see the clear correlation significance was coming from the root presence at the 30 to 45 and 45 to 60 and 60 to 75 in both the years, though there were no correlation in the initial stage in 2009-10, but still it is significant in 2010-11. The same pattern was also followed under optimally irrigated condition, but under root dry weight trait doesn't contribute to yield significantly, but still it have a very good positive correlation with the yield and root traits. The same data measured at 80 days after sowing. In this case, the early genotypes were approaching maturity. At 75 days after sowing in 2010-11, it was in the mid pod filling stage. At this stage, under non-irrigated condition, the root presents at 75 to 90 and the root present at 45 to 60, 60 to 75 and 75 to 90 showed a clear contribution with the yield significantly in both rootland density as well as root dry weight, and the consistent contribution where comes from the depth 75 to 90 in all the cases. As a summary, till now we have seen the depth-wise correlation. In summary, I just put the total root presence at the timing intervals or else the crop growth stages with the grain yield. The contribution where majorly there is no significant contribution were arrived at the 35 days after sowing, but the root started to showing a significant contribution to yield from 45 to 75 days after sowing through enhanced uptake of soil moisture. The contribution of root traits to grain yield is summarized as: at vegetative stage, root traits were showed positive contribution to grain yield in the initial soil depth and was consistent only at the 30 to 45 centimeter. At early reproductive stage, the contribution were positive in all the depths, but at the depth 0 to 30 to 75 had a significant correlation with the grain yield. At late reproductive stage, the significant contribution was shifted towards the deeper layer towards the deeper zones as with the increase in crop age. At this stage, the contribution was highly significant to the root present at 75 to 90 soil depth. When come to shoot biomass, at the vegetative stage, shoot biomass and SLA have a direct negative effect with the yield, but it was not maintained at maturity. Till vegetative stage, the shoot biomass and specific leaf area were negative, though it were coming in a positive correlation through LAI. It showed a direct effect with shoot biomass itself at the reproductive stage, but in the leaf area index, earlier it was showing a positive correlation as well, the higher direct effect to the grain yield, but at the reproductive stage it turns to become negative and caused a negative correlation with the yield. We can see the same pattern in an elaborated manner as we have measured the samples in different days after sowing compared to 2009-10, you can clearly see the shoot biomass and SLA were negative at the vegetative stage, and it starts showing the positive direct effect from 50 data of sowing. At this time, it was the initiation of pod had occurred in most of the genotype, and LAI showed a positive contribution at that time, and the SLA at 80 days after sowing showed a positive direct effect as well as significant contribution. The other aspect of the shoot biomass have canopy proportion and canopy temperature. It had the canopy proportion doesn't seems to contribute significantly with the grain yield, but the canopy temperature depression significantly contribute to grain yield at all the stages, showing their constitutive nature. At this stage, at 66 days after sowing, the direct effect was 361, and 70 it was 347, and these values are getting decreased with the crop age in both non-irrigated condition as well as the irrigated condition. The summary of shoot traits. In shoot biomass, the vegetative stage shoot biomass had no contribution to grain yield, in turn into positive and at reproductive stage. When come to SLA had a positive contribution across crop stages, and this contribution become highly significant at the mid pod filling stage with 0.01 level. LAI, it performed reversely as I mentioned in the previous table, but the CTD measured at reproductive stage, especially from 63 to 72 days after sowing, it showed a positive contribution to grain yield at 0.1% significant level. Beyond that, the CTD tend to become sparse and not significant as the crop approaching the maturity. While dissecting the yield components, when we talk about the phenology, it had a negative effect on grain yield, negative contribution to grain yield. As I mentioned, the genotypes that you have selected were very close in phenology, so we wouldn't expect much days to flowering positive relationship in this condition. But among the morphological yield components, harvest index showed a consistent significant contribution to yield, and shoot biomass have a significant contribution, but had a positive contribution but not significant to that extent. As well, the pod number also showed a significant positive correlation with the yield. When come to the analytical components, the P had a direct effect of 1.31 in 2009-10 and 0.968 in 2010-11. This was comparatively high than the reproductive duration and crop growth rate in both the cases. Under optimally irrigated condition, also the P showed a very good contribution to yield in both the years. The result summary of contribution of crop phenology and traits is both days to 50% flowering and days to maturity were negatively contributed to drought tolerant, both under non-irrigated and optimally irrigated condition. The shoot biomass produced, harvest index, morphological yield components were followed by contribute significantly to grain yield and followed by the shoot biomass, pod number and non-irrigated condition. All the traits except 100 seed weight were contributed to grain yield under optimally irrigated condition. When come to analytical yield components, among the components partitioning coefficient showed the consistent significant contribution to grain yield across irrigation treatment and years followed by the crop growth rate. The markers that we have used in this study contains 169 polymorphic markers out of 338 SNPs and 377 polymorphic markers out of 754 were used in this study to analyze the genetic diversity of these 10 chickpea genotypes. While constructing the genetic diversity was high in SSR markers and The polymorphic information content were also high in SSR markers, but the heterozygosity and major allele frequency was comparatively high in SNP and DOT. The dendrogram constructed using SNP markers showed a clear discrimination of drought sensitive as well as the low yielding genotypes compared to the drought tolerant genotypes. In SNP as well, in DOT, it also discriminate this drought sensitive genotype, and SSR also. So the objective was mainly focus on the narrow down the number of traits in chickpea based on their contribution, and also the using high positive path coefficient that reflected on significant correlation where the root length and root dry weight at reproductive stage with the consideration of plastic RDP rooting depth were significant in across stages, especially at reproductive stages. For shoot trait, the CTD performed consistently in both irrigated and non-irrigated condition. The LAI at the reproductive stage, LAI at the vegetative stage, SLA at the reproductive stage showed a clear significant correlation with the yield. Among the yield components, the pod number per area, the crop growth rate, and the partitioning coefficient showed a significant and also have a positive path coefficient effect. Therefore, the order of importance based on their consistency, robustness, stability, and heritability, we have ordered the traits as the partition coefficient followed by CTD and RLD. In worst come worst, we can consider RDW for the root strength and also for rooting depth. For yield components, we can rely on pod number per area and leaf area index. It also reflects in crop growth rate. The other traits to worth is the phenology and shoot biomass, which are the two important parameters that are very relevant and worth consideration. But in this study, the genotypic selection, as I mentioned, is skewed, and the phenology were not much wider, so it doesn't seem to contribute to the grain yield. Well, coming to the objective 2, it's just is evaluate to suitability of CTD as a trait and the crop and the stage of relevance for grain yield for grain yield under drought, to evaluate the crop stage of closest association and identify associated molecular markers. The plant materials were used consist of 5 highly tolerant, 53 out of 78 tolerant genotypes, 14 out of 39 moderately sensitive genotype, and 12 highly sensitive genotypes, and the irrigation was single, and the trial was planted in 14x6 alpha lattice design with 3 replications. The plot size were large enough, and the sowing date was 20th November. The measurements that were carried out in this experiment with the canopy temperature, especially at the mid-reproductive stage, crop phenology, grain yield, shoot biomass, and harvest index. For the relationship, we used one-way ANOVA, regression, and cluster analysis to cluster the genotypes based on the CTD performance as well as their grain yield. And also, 1849 markers data were assembled from Vashney et al. 2013 and used for association analysis. The relationship between CTD and grain yield was measured at the reproductive stage, started from 59 days after sowing to 82. There was a close association between the CTD and the grain yield was across all the stages. It was positive, and it became non-significant at the 82 days after sowing. Though all the regression values are positive, CTD measured at 62 days after sowing showed a significant explained 40% yield variation under drought stress condition. And based on this 62 days after sowing and 69 days after sowing sample, we used those data for the clustering, and also 73 days after sowing. Why especially these three, not in 59 days after sowing, is because these three samples have a high heritability compared to the 59 days after sowing. The CTD measured at 62 days after sowing explained yield critically was also compared with the previous year yield data, and it showed a consistent significance between the CTD and grain yield across years from 2008 to 2010. And these are all the clusters were done basically by using CTD at 3 stages, 62, 69 and 73, and grain yield at 2010-11. It shows the highest value of CTD yielded high compared to the high CTD and the lowest CTD. In other words, the genotype that have a lower canopy temperature can yield better that measured only at reproductive stage. But in this case, the canopy temperature was high compared to the ambient temperature, so it seems to be yield poor. The marker trait association was also carried out using for those 84 genotype using 1849 markers. It shows for CTD it gives 22 markers, but for the maximum number of markers were present for CTD measured at 69 days after sowing. It showed 10 markers, but the highest level of phenotypic variation was explained by the CTD that measured at 62 days after sowing. Though there were an interlink between the markers like TA14 and T13 that is also responsible for days phenology as well as grain yield was appearing with the CTD. And the number of markers were high. It shows the number of markers was high. It's showing that the CTD is driven by many characters such as stomatal conductance and the rooting performance and etc., phenology and etc. Result summary of this objective: CTD is considered to be a standalone trait that measures the plant water extraction constantly changing soil atmosphere continuum. CTD measured at mid-reproductive stage explained a major proportion of grain yield variation at a terminal drought, proving its worth as a proxy for grain yield. Though these associations tend to become very sparse when the increase in crop growth or it is approaching to the maturity. A cooler canopy temperature at mid-reproductive stage can be used as a selection criteria as it ensures greater grain yield under drought. There were large number of molecular markers that explained a major proportion of phenotypic variation in canopy temperature depression, 2 of them through phenology and yield as I mentioned. When come to objective 3, the root anatomy were compared chickpea with the other grain legumes and also between the chickpea types. The plant material and design crop management was separated into 2 experiments. The first experiment contains chickpea in comparison with the other grain legumes, and the second experiment contains the desi variety. To understand the difference between the desi varieties, the root anatomy of desi varieties with the cobbly ones. The trial was conducted in alpha lattice design, and the water level was maintained as non-irrigated condition. The root was sampled 2 cm piece, which is taken from the 10 cm above from the root tip, and were freehand sectioned and stained with Toledan blue, and image were captured under Olympus connected with a digital camera. The results show that compared to chickpea, the root diameter of pearl millet, pigeonpea and groundnut were smaller, but the cowpea have a large root diameter. The table shows the chickpea had a highest number of small xylem vessels as well as highest number of metaxylem vessels, contributing to the total number of xylem vessels were very high compared to the other legumes. Though the number of vessels were high, it have a reduced it have a very lower size of average xylem. Therefore, the total xylem passage of a single root was smallest in pigeonpea and groundnut and common bean, moderate in cowpea and chickpea, and largest in soybean. However, pearl millet has markedly small passage. These roots were represented based on the diameter for the comparison. While coming to the difference between the Desi and Kabuli, Desi had a very big stellar portion compared to very small stellar portion compared to the Kabuli, and they have a large cortex tissue compared to the Kabuli. And the major discriminating factor is the number of xylem vessels is moderately low compared to the Kabuli chickpeas, and also the size of the metaxylem, the diameter of the metaxylem vessels was also comparatively high in Kabuli. Therefore, the results summarize the xylem vessels and the number were apparently the most discriminating trait in legumes. The cortex of soybean and pigeonpea were relatively thinner than the other legumes. Chickpea and cowpea had a moderately high xylem passage per root, indicating that these are capable of absorbing water moderately and are well equipped for regular drought episode. The developmental cortical cell and steel tissue and their proportion is heavily influenced by moisture availability to the root system. The xylem vessels in Desis were fewer in number and narrower in diameter compared to the Kabuli types. Traits such as total number of xylem vessels, xylem vessel diameter, average xylem vessel size, and root cortex and steel ratio of chickpea varied between chickpea types and other grain legumes, which provide a clue on their drought adaptation. I would like to acknowledge my supervisor Dr. L. Krishnamurthy for his great assistance during my research work, and I also thank Dr. Vincent Vadas for his great contribution in funding, and Dr. H.D. Upadhyaya and Dr. R.K. Vashne and Dr. Mahendra Tudi for their great help in the marker data analysis as well as providing marker data. I also thank Mr. Subhay Singh for helping me in analyzing the data, and Dr. M. Vetrivendan assisted during my thesis writing. And I also thank Dr. Rosanna Mullah for extending my tenure to some extent, and I thank scientific and administration and supporting staffs, crop physiology, genetic resource department, and I also thank LSU, KSI, ICRISAT, JNTU, my university and library, and my family. Thank you.
Thank you, Purushottam, for presenting it so well. I'm sorry for the crammed presentation. The culprit is me. I wanted to bring down all that result that he has done with so much of effort to be presented. The end result is a crammed one. So this is for your questions, please.
Yes, sir.
Very good presentation. I have only one question. Why you say that CTD is a trait? It is the result of traits in the plant.
Yes, sir.
Maybe not somewhere plant anatomy or the soil water content. Why you say that CTD is a plant trait?
It's not actually, it is a proxy of trait. We can use it as a surrogate, but in this case we have mentioned as a trait.
And also another thing I saw was that your yields were
yes, sir.
I thought it should be a bit. I mean, what is your CTD? Is it canopy?
It's a canopy temperature depression.
Temperature minus.
It's not an actual canopy temperature. It is a calculated value of a canopy temperature with the difference between the ambient temperature and canopy temperature. It will show you the depression, how much the plant depresses.
So therefore, if the depression is more, the yield should be more, but your yield are coming down.
No, sir, the depression is more, the yield is more. The yield is
I think if
reduced.
If the canopy is cooler,
Yes, sir.
The yield should be more.
Yes, sir.
It all
Yes,
depends
sir.
how you calculate, I think.
Yeah, the graph that I have showed is the reversal what you have said. It inverts, as we said. The canopy temperature, if the cooler canopy, if the crop canopy is very cooler, it yield more. But if the crop canopy CTD value is high, it yield more.
So long as the canopy is cooler, then yes, we do.
Yes.
To continue the work on the roots, if you had 1 or 2 years more to work, what would you do?
I will use the large germ blossom lines, and I will concentrate to explore the anatomical part to find out any difference that can be easily done in the lab itself, that we can use it for high throughput. Yes, sir. Because under drought condition, the crop normally should have a moderate xylem vessels. It should not be large in xylem diameter, and also should have a cortical cells, high dense of cortical cells to regulate their water uptake under drought condition. Because we can clearly see in Kabuli, it is adapted to Mediterranean region. It have a large xylem diameter because it is habitat to grow in that well-watered region. So they can take more water at any time. No, sir. Yes. Okay. Yes. Because in wheat they have a
measurement of the conductivity will tell you that you have a relation between those xylem size and number difference and the capacity to change water.
Yes, sir.
In one of your slides, you showed a relationship between CTD and markers, which I
Yes, sir.
didn't exactly follow. But I think you were saying that there was a group of markers that was associated with CTD. That presumably mean different alleles of those markers discriminated the two CTD types or something like this. So to what extent does that reflect the intrinsic genetic difference between the lines that had a high and a low CTD, and to what extent was it associated with the CTD itself, and how would you get at answering that question?
So the selection itself, it's a diverse genotype, sir. We have selected 5 oil tolerant, and it's really 53 tolerant and 14 sensitive and highly sensitive 12 genotypes. So we have a we try to explore there is any genetic variation on the basis of CTD to have a high throughput drought tolerance measure. So we have tried it, and also we are interested to measure the association between the CTD as well as the market data.
Sorry, perhaps I didn't really explain myself well. There may be some lines which are very closely related to each other genetically, and others which are more distantly related. One of your early dendrograms showed that,
Yes,
something like this. So that means that the genetic variation is not evenly distributed in the sample, and therefore if you have CTD that is distributed in some way among the sample, you have to control for the uneven distribution of genetic variation. So I'm asking, did you think about how to address that problem?
the purpose of selection that I have mentioned in the first objective is, yeah, as you said, it was skewed population, but in this case the selection of genotypes is really diverse.
But are all of the lines equally distant from each other?
No, sir. It was purposefully selected to understand the variation between genotypes on basis of CTD.
So if you took the lines that had an odd number versus the lines that had an even number, you would have an association between the marker and the odd or even number, just because the genetic diversity is lumpy.
Okay.
Another question I have is that, you are talking about the number of xylem vessels and their thickness, the rooting depth, the root length density. They're all going to control the use of water.
Yes.
You may have thinner vessels, the rate at which the water will be utilized will be less. But how, one can really adjust these things for different environments. You may have soils of different water availability. You would like to regulate the use of water in such a way that you have more water utilized during the reproductive phase. But at the same time, you want to completely deplete the soil water also. Higher the use of water, more is the production. But at the same time, greater the proportion of water used during reproductive phase, you will have higher yield. So when you have different environments, you will really, I wonder how you will really optimize these things so that, the net result is high yield, And similarly, we say that we want to breed crop varieties for the drought environments. And again, the drought, every year it may not be a complete drought environment. You would like to have higher yield also. So more drought tolerant, lesser the yield in good years. This is not a question for you. This is, I think, a question for everybody.
Thank you, sir. Yes, ma'am.
How you really want to optimize the trade? Are we going to optimize the trades for different soil moisture environments, or we are going to follow an average path?
Yeah. Could I ask a question about the root sections?
Yes, ma'am.
You were comparing different species there, I think, were you?
The figure 3 you're talking about?
Yeah, that figure.
Yes, ma'am.
Different species are shown there.
No. It is not from the different species.
Okay,
it's different grain. It is different grain legumes in comparison with one cereals.
Compared with what?
One cereals, pearl millet, that I have shown in figure A.
Yeah.
3A.
Okay. But there was a difference between Desi and Kabuli, right? Which you talked about.
Yes, ma'am.
Which is in the top there.
No. The first horizontal 3 are Desi, and the image DEF is Kabuli.
Yeah.
These three are Desi and
Yes.
these three are Kabuli.
Yes, and the point was that Desi. And so are those different genotypes of Desi and those are different genotypes of Kabuli? Is that what you're saying?
Yes, ma'am.
Okay, that's fine.
If there are no questions, thank you for this thing. About the material that is used for CTD. We have had the whole reference set of chickpea germplasm categorized into various groups, and from each group we tried to take material. There were about 78 entries which were used for that, and nevertheless, yes, the genetic lumping you were just talking about is quite possible, because we have heavily skewed material brought in, and we are very sure we wanted to just go for a preliminary evaluation of the whole thing. Now we know a marker which is appearing at 62 days doesn't appear again. And we have quite something like 24 markers that are appearing, but at a given time, only one stage you will find large number of markers. So I suspect that it is all co-linked with either phenology or yield formation or many other things, because we know the drought tolerance, one trait shifts after other traits, and it comes for the trait this thing. And we had this question of optimization of the soil moisture. Whatever the efforts we do, ultimately it is to use all that moisture. The jugglery that we do, all that we talk about, and the problem has arisen that without any genetic conclusion or agronomic conclusion, that he is trying to prove a point, that which is the trait which is very important, what is the order of the traits that we have to take, shows that it is necessary to work on that. And in the process, you might have seen a genotype which is flowering at 37 days, which just had utilized about 120 millimeter of available 230 millimeter of soil water. And then there is another genotype which uses close to 150 and going up to 50 days to flower. We have used that. And if you go beyond that, it is futile, we know that. So it seems the seasons that Purushottam has taken the sample, that 50-day-old flowering time, one has utilized all that. But as you have rightly said, we don't have any solution for controlling the drought, so we will not be able to do that. So I think the question that you have asked about root anatomy, the Kabulis and Desis, they originate from various regions. And then the growing condition, soil moisture varies. And the wheat example that we have taken and just started making an effort to find out if the anatomy can make a good root phene. That's where we have to finish it. And we all have to be little merciful, and the work is such a heavy one, and we can't expect that this much from a PhD student. So nevertheless, we did it. And I will say, thanks Purushottam for this nice presentation, and thanks for all of you to come and just witness this presentation. Thank you.